Like buried treasure, they lie hidden from sight. Echoes of an ancient past, they whisper secrets and tell tales once lost to time. Fossils are remnants or impressions of ancient organisms that are naturally preserved in stone. While there are hundreds of fossil types, they are often grouped into two major categories: body fossils, which are the preserved remains of plants and animals, and trace fossils, which are records of an animal's behavior, such as footprints. Together, they form the fossil record, a primary account that tells the story of life on Earth through stone. Fossilization, or the process of preserving organisms in stone, can occur in countless ways. These methods are largely grouped depending on whether the organisms are altered during the fossilization process. Fossilization that does not alter a specimen can help to preserve its original form and texture. Among many methods, this group includes organisms that have been frozen, preserved in tar pits, and mummified. One special case involves trapping organisms, oftentimes insects, in amber. This process begins when an organism is covered in tree sap. The sap, or resin, forms a protective seal around the entrapped organism. Over time, the soft resin hardens and turns into amber, with the organism suspended within. This process creates a biologically inert tomb for the organism, allowing its soft tissues to be remarkably preserved. Other fossilization methods change the specimen as it is being preserved. For instance, carbonization transforms soft tissues into thin black films of carbon. In fact, countless layers of carbonized plant material create a well-known fossil fuel: coal. But one of the most common types of fossilization that changes a specimen is called permineralization. Permineralization begins when minerals from water or the ground enter the pores of dead plant or animal material. Over time, the minerals attach themselves, clinging onto cellular walls and building a crystalline network in the empty cavities. This mineralization hardens the bone and turns it into stone, thereby preserving its original structure in fossil form. When conditions are right, fossilization can preserve crucial information about an organism. Permineralized wood often contains enough information to identify its tree genus and sometimes its species. Insects encased in amber have been so well preserved that their genetic material was extracted and partially sequenced. And footprints left behind by ancient hominins help paint a picture of what life was like for early human ancestors millions of years ago. With every fossil uncovered, the planet's ancient past becomes clearer, helping shape our understanding of our world today. Jellyfish can be found all over the world, from deep oceans to shallow coastal areas. They've been around for hundreds of millions of years. Boneless, brainless, and bloodless, jellyfish are some of the most diverse and fascinating creatures in the sea. Jellyfish aren't actually fish. With thousands of different species in 2 different biological phyla, jellyfish is more of a broad term than anything else. Unlike fish, which have backbones, jellyfish are invertebrates. Jellyfish got their common name from the jelly-like material they're made out of, called mesoglea. In recent years, scientists have started using the umbrella term sea jellies to clear up the confusion. There is an immortal jellyfish. Jellyfish can reproduce sexually by releasing sperm and eggs into the water, and reproduce asexually by splitting into 2 or cloning. But at least one jellyfish can actually reverse the aging process. The Turritopsis dohrnii has earned the moniker the immortal jellyfish for being able to undergo a process called transdifferentiation. An adult or a juvenile under stress, instead of dying, can revert back to a polyp and begin the life cycle all over again. It is the only animal in the world that is known to be able to reverse its life cycle.
$385 million years ago, the land was all but empty. The continents were home to only some sparse plants and fungi and a few pioneering arthropods. Life in the oceans, however, was booming. The waters teemed with squid, bony fish, and sea scorpions. But there was one group of fish that was about to go its own way. In time, these enterprising fish would undertake one of the most important journeys in the history of life. They'd follow in the footsteps of the arthropods to become the first vertebrates to live on dry ground. But they couldn't just move on to land. Not yet. First, they had to acquire the ability to breathe air. The giant link between fish that breathe through water and fish that breathed air has been really hard for us to study and understand. That's partly because the organs that did the work of breathing, either gills or lungs, don't fossilize well. But it's also because until pretty recently, there just haven't been a lot of fossils from that time for us to study. Fish began their transition onto land about 400 million years ago, during the Devonian period. And for a long time, one of the few fossils that experts had to work with was a fish named Eusthenopteron, originally found in Quebec in the 1880s. It lived in shallow, estuary waters around 380 million years ago, and like the famous coelacanth, it was a lobe-finned fish. Instead of having long, delicate fins with lots of joints in them, like you'd see in a goldfish, its fins were shorter and stronger. These hardy limbs could have helped Eusthenopteron move itself along in the shallows as it hunted other fish, but its fins probably weren't strong enough to let it walk on land. Another major find came about in the 1930s, when scientists uncovered Ichthyostega, a meter-long creature found in rocks in Greenland dating back 364 million years. Ichthyostega had a body that was a lot more salamander-like, including a fully developed pelvis, strong limbs, and even fingers. But Ichthyostega still probably dragged itself around with its front legs, a bit like how a mudskipper moves today. And it also had a thick paddle-like tail for swimming, which means it likely spent a lot of its time in the water. But perhaps neither of these fossils is as key to this story as Tiktaalik, a 375 million-year-old animal found on Ellesmere Island, Canada in 2006. Tiktaalik had a fishy body, but a head like a salamander's, and stiff, leg-like fins that could have supported its weight outside of the water. And most importantly, it also had bigger primitive hips, so its hind limbs had something to anchor onto, an important step in becoming a fully four-legged animal. However, Tiktaalik's travels on land were probably still stuck in front-wheel drive. Even though its legs and hips were bigger than Eusthenopteron's, they weren't strong enough to bear the strain of walking on land. With With traits that seem halfway between a fish like Eusthenopteron and a four-legged animal like Ichthyostega, Tiktaalik is a textbook example of a transitional fossil from this time. And despite their differences, all three of these fossils are considered to be tetrapodomorphs, a group that includes early four-footed animals and the lobe-finned fish that are closely related to them. Now, one big trick to living on land, of course, is breathing. So how did these animals, which had been adapted for millennia to life underwater, start to breathe air? Well, we know that all three of our friends, Eusthenopteron, Tiktaalik, and Ichthyostega, had gills. Because even though gills themselves don't fossilize very well, the bony arches that support the gills do, and each of these animals had gill arches. But even though they still had gills, that doesn't mean they couldn't breathe air. It turns out the most important clue for when fish started breathing air isn't the absence of gills. Instead, it's the shape and location of a little hole in the skull. This hole can still be found in many fish today. It's the opening of a tube called the spiracular tract that's used to bring water in towards the gills. This feature is really handy because it allows fish to breathe when their mouths are busy eating. And this little skull hole can tell us a lot about when tetrapodomorphs first became able to breathe air. All you have to do is compare where it shows up in the fossils with where it appears in different kinds of modern fish. Now, in most modern fish, the opening of the tract appears on the sides of the face, near the front of the skull, which puts it pretty much right on top of the gills. But there are also fish today that breathe air, and in one of these fish, the bichir from Africa, the hole is bigger and sits on the top of its skull, farther back, kind of like a blowhole. What's more, the opening also sits at an angle, not straight up and down. That's an important clue because this angle creates a more direct path for air to travel to the bichir's lungs. Now compare all of that with the skull openings in tetrapodomorphs. In Eusthenopteron, the spiracular tract opens up near the front of the skull, just like in most water-breathing fish with gills. But the hole is on the top of the skull, not the side. In this way, it kind of resembles the bichir, which uses its head hole to breathe air from the surface of the water. So Eusthenopteron was maybe breathing air in addition to water, and since the hole was close to the front of the face, they probably used their gills to handle both. But in both Ichthyostega and Tiktaalik, that hole is much bigger, is closer to the back of the skull, and sits at an angle, just like it does in the bichir. This means the tract in these ancient animals was pointing right to where a primitive set of lungs would have been. It's not the clear-cut evidence that a nice set of fossilized lungs would be, but having a larger passageway that sits closer to where the lungs are in today's lunged fish is a good sign that both Tiktaalik and Ichthyostega had some sort of primitive lungs. As for where these lungs came from, experts think they derived from an organ that many modern fish still have today, a swim bladder. Swim bladders are often filled with air, which fish gulp down to help keep them buoyant, and ancestors of Tiktaalik and Ichthyostega probably had them. The thinking is that among some lobe-finned fish, the swim bladder became bigger and contained more blood vessels, so it became better at putting oxygen into the bloodstream. In time, this organ took on a different function, finding a new use for the air that was already there: breathing. This kind of makes sense. If you look at air-breathing fish today, like the lungfish and the bichir, their swim bladders are split into two and so full of blood vessels that they look basically just like our lungs do. But why would any fish, modern or fossil, bother with breathing air when there's all that lovely water around? Well, during the Devonian, things were complicated, and all of those complications led to a steady drop in the amount of oxygen in the oceans. On land, new plant species were diversifying, which you'd think would be good for oxygen levels, except all of those new plants were also dying on land, and then getting washed into the ocean. All of that organic material fueled huge blooms of algae, and then bacteria, which in turn sucked up oxygen from the ocean. So by the late Devonian, right when fish started to transition onto land, oxygen in the air was really low. Some estimates go as low as 13%, compared to almost 21% today. For animals, low oxygen is generally bad, but for organisms that live in the water, it's even worse, because oxygen concentrations are always lower in the water than they are in the air. So when oxygen levels first started to fall 385 million years ago, the benefit of transitioning onto land was pretty clear: no more gasping for breath in the water. Being able to breathe air made Tiktaalik, Ichthyostega, and maybe even Eusthenopteron more energetic and better able to hunt their food. And these first air breathers eventually gave rise to the true tetrapods, the first vertebrates to live on dry land full time. Eventually, they lost their gills, and by the time the Devonian period had ended and the Carboniferous was underway, they had also lost their sporacular tracts and started using a totally different kind of skull hole for breathing, called nostrils. But those lobe-finned fish that were our ancestors weren't the only fish that figured out how to breathe air. Today there are lungfish, mudskippers, bowfin, and bichir. They're not direct descendants of the likes of Tiktaalik. Instead, they each acquired the ability independently at different times. It just goes to show you that breathing air has turned out to be very convenient for a lot of us over time. So remember, for all the breaths that you're taking today and the way that you're taking them, you owe your fishy ancestors a debt of thanks.
Charles Darwin spent 5 years circling the world to discover what became the origin of species. Alfred Russel Wallace spent 8 years traveling through South Asia to witness the story of evolution. But you can discover the same thing today without going anywhere, because the story of evolution is right there in your body. Your tailbone used to be a tail. If you can move your ears, that's thanks to muscles you share with other mammals. That pink bit in the corner of your eye, a leftover of a third eyelid we can still find in lizards, birds, fish, and others. Goosebumps are a feature we share with lots of animals that didn't lose their body hair. And even hiccups trace back to the brains of fish and tadpoles, a brain signal they use to squeeze water through their gills. These body parts are just a few scattered pages in a larger story about our evolution. But there's other fossils hidden inside our bodies that tell an even clearer tale about how we came to be the way we are. One of these stories is so simple it can be told with a single picture. These are images of chromosomes, the big chunks of DNA inside every one of our cells. When we look at images of human chromosomes next to those of other apes, they line up nearly identically, except for human chromosome 2. It seems to have no match next to the other ape chromosomes. Even weirder, humans have 46 chromosomes in total, 23 pairs. Those other apes, they have 48 chromosomes, 24 pairs. What could explain this? If we're supposed to be related, why are we missing a chromosome? Turns out we aren't missing a chromosome at all. Sometime during our species history, those 2 chromosomes that are still separate in other apes were welded to become our single human chromosome 2. Us, chimps, gorillas, orangutans, all inherited the DNA we have today from some common ancestor with 48 chromosomes. But 2 of ours got stuck together along the way, and you can even see how they match up. If 2 chromosomes were glued together, you'd expect to find 2 centers, or centromeres, and we do. We'd also expect to find telomeres, the ends of each original chromosome, stuck head to head in the middle, and we do. Thanks to sequencing the human genome, we can spot the exact DNA base where this fusion happened. 113,602,928 bases from the end. A fossil of evolution hidden in our own bodies. In Celtic mythology, there's a place called Tir na Nog, a land of eternal youth. When one scientist discovered a protein that keeps stem cells forever young, he named it after this place, Nanog. The human Nanog gene is located on chromosome 12, at least the version of the gene that works. There are 11 other broken copies of Nanog spread throughout our DNA. How did these extra copies get spread through our genome like lost socks? When DNA is replicated, sometimes the same bit gets mistakenly copied twice. And sure enough, we see one broken copy right next to the working nanog gene, like a messed up DNA photocopy. We call these broken genes pseudogenes. And what about those other 10? There's another way these pseudogenes can spread. Nanog, like every gene, is copied from DNA into RNA letter by letter before that RNA is used to build a protein. But every once in a very long while, that RNA gets mistakenly copied backwards back into DNA, pasting a new broken version of the gene somewhere in our chromosomes. After searching the billions of letters in our genome, we've found 10 of these lonely nanog leftovers in our DNA. Their locations seem random, like they were blindly tossed in. Now, say you were to read another animal's genome and you found these very same broken copies of nanog, each pasted in the very same locations among billions of letters of DNA. That would be a heck of a coincidence, unless it wasn't a coincidence. This is what we see when we search for nanog in a chimpanzee's genome. The original and 10 of those lonely broken copies, all in the same locations as ours. This only makes sense if millions of years ago, you and me and the chimpanzee share an ancestor whose DNA was already littered with these broken genes. We all inherited this pattern because we share an evolutionary link. Of course, it's one thing to share a link with chimps, but chickens? That would require some strong cluck and proof. One thing you have in common with a chicken, you both came from an egg. Inside a chicken egg, the growing embryo produces something called a yolk sac. It surrounds, well, the yolk and helps gradually absorb nutrients to build a baby bird. But human eggs are incredibly small, only about 1/10 of a millimeter across. Unlike birds or turtles, our eggs don't make a yolk or even a shell. We get our nutrients and protection from our mom's body. So you'd think no yolk sac. But that's not the case. Humans make an empty yolk sac, and it disappears halfway through pregnancy. See, this is a clue that our eggs and bird eggs and even reptile eggs all share an evolutionary link. But can we prove it? There's an essential ingredient in egg yolks called vitellogenin. It's a protein, which means there's a gene whose DNA codes how to build it. Egg-laying vertebrates all have at least one copy of this gene. But what about us? If we really are descended from animals that once laid eggs full of yolk, can we find some leftover of that yolky gene hidden in human DNA? In 2008, researchers found just that. Fragments of once active egg yolk genes hidden in our own genome. And when they compared our DNA to the chicken versions, they even had the same neighboring genes around them. This is exactly what evolution would predict. Just like fossil bones, we see remnants of our ancestors broken and buried in these hard to find places. There are many myths of creation around the world, and most agree on one thing. Humans are special, and we need a special story for how we got this way. When we take a close look at our own biology, we can see that special story is evolution, and it's a story we share with every other living thing on Earth, some more than others. To some people, the idea that humans, our bodies, our brains, our thoughts, are also the products of evolution removes some of the magic of being human. But it doesn't have to. When Darwin wrote, "There is grandeur in this view of life," he meant that being a creature molded by evolution is a totally different kind of magic. The kind that still amazes you even when you know exactly how the magic is done. Stay curious.
Birds come from a long line of survivors. It started millions of years before the asteroid strike with a dinosaur family called theropods. The theropod family featured reptilian specimens, some of which had bird-like traits, including feathers and three-toed feet. A distant cousin of theropods and today's birds was the Archaeopteryx. Fossils from Germany date the animal to about 150 million years ago, and they show how the animal may have even had both reptilian and avian traits. Many ancient birds would also exhibit this mishmash of traits. But eventually, some of these avian dinosaurs evolved to have more recognizably bird-like characteristics. One example is the sparrow-sized Sinornis santensis, discovered in China around 1990. The archaic creature is considered one of the first birds capable of flight. In 1992, a new specimen was discovered in Antarctica. Called Vegavis ayi, the 67-million-year-old creature may have looked like a duck and sounded like one. The fossil included a vocal organ called a syrinx, which is only found in today's quacking waterfowl. About 1 million years after the time of Vegavis ayi, the asteroid struck Earth and redirected the evolutionary paths of many animals, including birds. 3 bird groups of the theropod family managed to make it through such a cataclysmic event, and scientists are still uncertain as to how. Ever since, birds have been able to adapt and evolve, eventually blooming into the over 10,000 species alive today. So the next time you're looking out at your bird feeder, remember that you're looking at the modern-day dinosaurs.
Despite what you may have heard or thought, fur and hair are actually the same thing. We just use a different word to describe the fur that we have, but they're both the same kind of pelage or hairy body covering. And fur is one of the defining characteristics of mammals. And while we're not sure exactly how it evolved, we do know that it's super old. Fur is thought to have evolved as a way for animals to keep warm by trapping air against the skin, which prevents heat loss to the surrounding environment. And today, it also has a lot of other functions. In warmer climates, fur blocks the sun's heat and UV radiation from reaching the skin. It can also act as camouflage, make animals appear larger when they're feeling threatened, and certain coloring patterns can even help keep bugs away. But fur can also make it more difficult to cool down. One way many animals lose heat is through panting or taking short, forceful breaths. When animals pant, heat from the insides of their mouths evaporates into the surrounding air, which cools down the blood in the veins of their tongue and cheeks. This cooled blood prevents their brains from overheating. But in hot climates, panting can't always keep up with how hot it gets, so many mammals have to rest in the shade during the hottest part of the day. To avoid this, some mammals also sweat a little. As sweat evaporates from the skin's surface, it takes some of the body heat with it, increasing the animal's ability to lose heat. But lots of sweating comes at the cost of losing more water, not really what you want if it's already hot. Plus, if sweat soaks the fur, then heat can't really escape anymore. So for most mammals, fur combined with panting, shade, and a bit of sweating usually cools them down just fine. But why don't we have fur? Why did our lineage develop super sweaty bare skin exposed to the elements only to cover ourselves up again anyway? The answer might actually lie in another aspect of human oddness, the evolution of bipedalism, and often one big change can lead to another. When our early relative Australopithecus came onto the scene in East Africa some 4 million years ago, we started to see major changes in how hominins moved around. Fossils of hip bones, femurs, and foot bones show that these hominins were able to walk on 2 legs, but the bones of the fingers and shoulders show that they also spent some time in the trees. It wasn't until our genus Homo emerged around 2 million years ago that we became fully committed to walking bipedally. And around 1.8 million years ago, Homo erectus took it a step further. Its tall stature, long limbs, and bowl-shaped pelvis, which we humans have today, gave Homo erectus a more ideal running body compared to those shorter, stockier hominins that had come before. And some scientists think this ability to run allowed Homo erectus to hunt using an uncommon method called persistence hunting, or chasing prey until it collapses from exhaustion. In fact, by calculating the amount of water that humans lose when they engage in persistence hunting, a group of anthropologists recently found that Homo erectus could have hunted for over 5 hours straight without needing a water break. And we carry on that legacy today. We're the only living primate that can engage in persistence hunting. And I say we loosely because I definitely can't do that, but it is possible. Now, for Homo erectus and later hominins who may have been persistence hunting on the open savannas during the Pleistocene epoch, they could have been at risk of overheating. And it's this connection between how we move and how hot we get that has led many scientists to suggest that our locomotion was connected to our loss of thick fur. Hominins with less fur could sweat more efficiently, which would cool them down much faster without having to take breaks in the shade and lose valuable hunting time. Until recently, though, these experts found themselves in a chicken and the egg scenario. Did we lose the fur first, or did we start running first? Was Australopithecus hairless, or was Homo erectus still hairy? This is where fossils aren't much help anymore. So rather than trying to figure out when being furless would have been beneficial, a group of scientists tried to figure out when fur would have still been necessary for survival. Remember that fur is a great insulator. Even mammals living in hot climates have fur, which comes in handy when the temperatures drop at night. By looking at the environments Australopithecus lived in and how many calories they probably consumed and lost in a day, these scientists found that they couldn't have survived being hairless at night. Without controlled fire, which doesn't show up in the fossil record until millions of years later, they just wouldn't have been able to generate enough heat to keep up with what they would have lost without fur. So this tells us that Australopithecus probably still had a considerable amount of fur until they disappeared from the fossil record around 2 million years ago. This means that extensive fur loss occurred at some point within our genus, Homo. And DNA evidence from our own skin can help us pinpoint when that happened. Human skin comes in a variety of shades, which are thought to reflect genetic adaptations to UV radiation from the sun. Darker skin is better protected from this radiation than lighter skin. That's why many people with ancestry from places near the equator, where the sun strikes the earth at a higher angle, have darker skin than people with ancestry from farther away. Now, this protection wouldn't have been necessary if we had fur, because fur acts as a barrier to UV rays. We can even see this in other primates. Under their fur, their skin is lightly pigmented, but skin that's regularly exposed to the sun becomes darker over time. This means that if a hominin species did have dark skin, it must have already lost its fur. One study published in 2004 showed that a gene variant associated with dark skin called MC1R already existed at least 1.2 million years ago, suggesting that at this point in our history, hominin skin was adapted to intense sun exposure. And who was already walking around Africa 1.2 million years ago? Good old Homo erectus. The individuals with naturally thinner fur would have been better able to cool down, allowing them to run and hunt for longer without needing to rest as frequently. And these more successful hunters would have passed on their genes more often. Over time, fur would have become less common, until eventually the species was naked. So bipedal running and fur loss are closely connected. Both allowed us to become successful persistence hunters, which drove further fur loss.